Hi, Joe.
Hi, Aaron.
How are you doing?
Good.
Thanks for coming.
Really nervous.
Oh, don't be nervous. Yeah, don't be nervous. It's gonna be great. Do you want to test out your slides?
Sure. That work?
Looks good. Yeah, looks really good.
Okay. Let me try a video and
Yeah,
see if I have
sure.
a video. So very. So the network is fine.
Yeah, it looks good.
Great. Okay.
Hi.
Hello.
Hey. Um, do you want to test your slides, Weiguang?
OK,
Thanks.
test 啊。
Aaron,
Hey, it's long.
hey Drew, Weiguang,
Hi,
morning.
morning.
Have you tried slide? Have you tried slide here?
He's about to.
Okay.
OK,
Oh, Joe, I invited Yang to my party at my house last week.
Oh,
Yeah, she came to it.
how is it going? How's the party?
The party was amazing. I have a swimming pool, barbecue at my house.
Is the swimming pool usable still? I saw the big closed by no fence.
Oh no, the fence is just so people don't run in and drown. You have to
Oh.
in California. You have to do it, but the
Alright,
pool
sure.
is heated. It's really nice.
Great. Okay.
I haven't
Let's put
seen her
it.
for a long time.
sorry.
Yeah.
Right now, I'm in a quarantine hotel in Taiwan.
Oh. Okay.
I went to Taiwan yesterday.
But they're still in quarantine in Taiwan.
Just 3 days,
Oh, okay. Okay.
but it's like 1,000 times better than Shanghai. It's so pleasant. It's just they gave me nice fruits, nice taxi ride, big room.
Okay. Yeah. That'd be nice.
Yeah. Next week I have to quarantine in Shanghai. It's gonna.
Oh,
Yeah.
use a nicer hotel than last time.
Yeah, we'll try. So wait on.
Is it OK?
No, switch the.
You swap the display.
OK.
Yeah. You use a two screen, right? So we have to.
Yeah.
I don't know whether it's about Yang or something. So but the sleep research people has going exponentially increasing in China, all over the institute. I met several people in not just Taiwan, also in institute. Weiguang is in Fudan. Maybe you should visit this time. They got a very new.
Okay. Yeah. Me?
Overturning young people. Yeah. I think you should visit.
Yeah.
Yeah.
They are just opening last year.
Okay.
Physically opening.
Yeah.
I'll come in September 10th. I'll be in Shanghai.
Great.
Great. Yeah, try to slideshow.
Did it work, Weiguang?
Not yet.
So when you
Yeah,
Okay.
you just click the swap display.
no first
buttons.
first click play. Yeah, that's good.
Yeah. Okay, it worked. Okay.
It's okay. Is it
Yeah,
okay? Okay.
Mhm.
Thank you.
Yeah. Okay, this way.
Okay.
Well, there's another postdoc from Zhang's lab, who is a UNC, working on some sleep and neurogenesis. Is coming interviewing
Oh wow.
somewhere in Shanghai institution. So yeah, it's coming.
I've been
You-
helping out with interviews for Westlake University. There's a bunch of sleep people coming through.
Really? Okay. You met Zhang in Golden Conference, I believe.
Yeah.
Song Zhang Song, yeah. Mm.
Yeah, it seems like Yang is interested in microglia now.
Yeah.
Sleep instead of still dissecting the sleep circuits. Yeah.
Well, with microglia.
Yeah, I think so. I think also some ongoing other project.
Yeah.
Like the growth hormone or something.
Mm-hmm. Can you guys get to SFN or not yet?
This year?
This year?
No, maybe not.
No.
Yeah, no.
Maybe in 2 years.
Oh, 1 year, next year should be fine. I firmly believe.
That's what you said that same thing last year.
Yeah, I always believe next year we will be fine for everything.
Yeah.
Don't have to wait too long.
Yeah. Yeah, these in-person meetings have been great. I've literally been traveling all around the world all summer.
Yeah. So where will you visit in Taiwan?
The Taiwan SFN. It's in and then I'll go to Taipei. Yeah.
Okay.
It's like their annual meeting.
Mm-hmm. Is it somewhere in Taipei?
It's in Linkou, which is like in between the airport and. Actually, I don't know where it is, but it's not far from the airport.
Okay.
But my main plan is to go around Taipei a lot.
Mm.
I already made reservation for the seafood hot pot restaurant, and then other things.
Cool. There's a lot of nice food
Yeah.
to try.
Yeah. How about
This is academic Seneca? It's kind of like very like our campus.
yes,
I had many friends there.
yes. How about in-person meetings in Shanghai? Are they back?
We just had a small 50%
Okay.
meeting
Okay.
hosted by Fudan. Yeah, it's great. Yesterday, so I've been there for a half day. Nice. We have a discussion. Yeah, it's quite a while.
Okay.
We has the 1,000 people meeting next month, and the CNS meeting will be there last year. So in Suzhou, so I think it will be fine.
Okay.
I'll be there for a couple of days. It has some threshold. Some meeting like over 50 people, 100 people, or something. So I hope the 1,000 people meeting will be fine.
I want to do our 200-person ALS meeting some point.
October, yeah, it was after a while.
Yeah, we'll see.
It will be 200 person. So many.
Well, we'll see. Well, we'll see.
Okay.
We'll see what the regulation is.
Okay. Should we get it started?
Yeah, that sounds great. So,
Sure.
hi everyone. Today's a Taiwan edition of NeuroZoom, broadcasting from Taiwan. Next week, because it's Labor Day in America, we will take a week off from NeuroZoom, but then we'll resume the following week, September 12th. So tune in then. Let me and Zhang know if you want to present your work. So it's really a pleasure to introduce our first speaker today, Dr. Zhang. She's an investigator at the Institute of Neuroscience in Shanghai. She received her undergraduate degree in Wuhan University, then went to Imperial College London to do her PhD. And here she had a very interesting paper in Nature Neuroscience where she explored the question of if sedatives that people take for sleeping that are used to induce sleep, if they engage the same or different pathways in the brain that natural sleep engages. She then came to Berkeley, where she did a postdoctoral fellowship with Dr. Yang Dan, and here she had this really amazing paper in Cell, which I teach to genetics students, neuroscience students at Stanford, and here she really solves a like a century-old problem on it's about sleep center and sleep active neurons. She uses a wide range of techniques from screening, activity-dependent labeling, gene profiling, chemogenetics, the everything, and she defines a certain region of the brain that acts as an excitatory sleep center, and it really changes the way we think about sleep and sleep regulation. And since that paper, she's had her own independent laboratory in Shanghai, and she's going to tell us what she's up to. Go ahead, Zhe.
Okay. Hi everyone, I'm Zhe. Really thanks for Aaron's introduction, like about all the way from my PhD to postdoc. Moreover, thanks for the invitation from Aaron Zilong for the opportunity to give this NeuroZoom talk. I really appreciate the chance to introduce my lab and our recent work. My lab focuses on the dissecting the neural circuits for maintaining the homeostasis of the body function. We start a range of the functions, such as sleep, just Aaron mentioned, and respiration and many more. Today, I'm going to talk a project that we recently published regarding the thermoregulation and some extension of this project that is currently ongoing in the lab. We all know that the endosomes, such as human, have really stable body temperature. This offers some advantages, such as stable and efficient metabolism, and some says it could serve an immune function by preventing from certain infections. The maintenance of the body temperature is tightly controlled by the brain. Early study identified that the temperature information can be sensed by various temperature-sensitive ion channels in the skin, and then send the information into the brain to control the activity of the tissue function, such as the brown adipose tissue and skeletal muscle, to produce more or less heat, as well as the blood flow for the heat dissipation. Several brain regions have been identified to be important for the thermoregulation, including the dorsal-medial hypothalamus, preoptic area, and the PB. More recent research started to reveal the fine details of these neural circuits of the thermoregulation, specifically by identifying the cell types in each brain region that are responsible for controlling the body temperature. This graph summarizes the previous identified cell types. You can see there are many more red than the blue, means that the cell types responding to the code information are much less well known. On the other hand, the code-sensitive neurons should be very important for the thermoregulation circuits, since activating the thermogenesis in response to the code is essential for the animal to survive from the cold environment. Therefore, in order to understand the neural circuits for the body regulation better, we are trying to search more cell types that are cold sensitive in the brain. We start out using a systematic approach to search for the brain regions activated by the cold temperature by using the functional magnetic resonance imaging. This is a collaboration in our institute with my colleague Zhang, who is an expert in perfecting the FMI technology and its application. Okay, thanks to his help, we're able to record the bolt signal from the mice with good resolution and stability. And we are able to combine this bolt signal with a water jacket we made with alternating hot and cold water to change the environment temperature for the mouse. This kind of experiment allowed us to screen the whole brain for the regions that are artificially activated by the cold environment. When we qualified the results, we saw a brain region called the ventral medial hypothalamus that showed highest activation by the cold. So having found the cold VMH being cold responding brain region, we then cross-validate our results using a different technique. We examined the C-force expression after putting the mouse in the cold environment for 3 hours. Consistent with our FMI data, we found a robust C-force expression in the VMH after cold stimulation. So that's why using fMRI and C-force staining, we are able to find VMH to be a brain region that responds to the cold environment. Interesting, this region is not part classic thermoregulation pathway. We next question we would like to answer is the specific cell types in the VMH that are cold activation. We label the cold activated neuron in the VMH using the paradigm I just described and co-label with excitatory or inhibitory neural markers. We found the majority of the C-force positive neurons are Weiguang positive, which means the cold sensitive neurons in the VMH are excitatory neurons. Okay, to further narrow down the cell type of the cold responding neuron in VMH, we analyzed the single cell transcriptome data from the excitatory population. We found the excitatory population can be divided into 2 sets of markers. One group is pre-dynorphin positive, another one is pre-dynorphin negative, but expressing PACAP and TAC1. This analysis allowed us to using these markers to genetically label this subgroup of excitatory neurons in the VMH. We first tried to use the same C-force co-label method to see which subtype shows cold responding phenotype. We found that pre-dynorphin positive neurons are preferentially enriched with the cold activated neurons. Now we have the genetic marker for the cold sensitive neurons in the VMH. We can do more precise recording of their activity during the temperature change. We adapted our water jacket from the FMI experiment, but using the fiber photometry to measure calcium signal from the pre-dynorphin positive neurons in the VMH. Consistent with our FMI data, we found a very robust activation of these neurons by cold temperature. You can see even with a single trial raw data, you can see the calcium signal change with the temperature. And we combine the data with multiple mice and experiment. We saw a robust activation during the transition to the cold temperature and the deactivation during the transition to the hot. We then wonder whether this activation of these neurons is responding to a relative temperature difference, or do these neurons have some favorite temperature? So using the same setup recording technique, we modified our experiment by using a stepwise temperature change to test this 2 possibilities. If the neurons are responding to a relative temperature drop, we should see an activation during each step of the transition. On the contrary, we found the activation only when the temperature dropped below 10 degrees, suggesting that these neurons are truly responding to the cold temperature. As a control experiment, we also record other cell types like the Weiguang, TEC1, and PACAB, the other cell types in the VMH using the same setup. None of these subpopulations showed activation to the cold temperature. As previous studies that identify the VMH always shows some like sex-specific activity. So I wonder if the temperature sensitivity to these preadenorphin positive neurons in the VMH is also sex-specific. When we analyze our data separately in male and female mice, we found a robust temperature-dependent activity in both sexes and no difference between them. We further test whether the preadenorphin positive neurons in the nearby regions, like in the hypothalamus, showed sensitivity to the cold temperature, but found that not. We further test whether the fever could activate in the preadenorphin positive neurons. Since the fever is always associated with enhanced thermogenesis and feeling cold, which is similar to the response to the cold environment. So IP injection, the LPS really induced the fever, but contrary to our expectation, LPS induced the fever did not affect the activity of this neuron. Taken together, this data indicate that the preadenorphin neuron, positive neurons in the VMH are cold sensitive neurons with a really high specificity. And we next asked whether this neuron could directly regulate the thermogenesis. To this end, we express this step function opsin SSFO in the preadenorphin positive neurons in the VMH. Use a one-shot laser stimulation, this opsin can stay open for half an hour, allow us to test the effect of the activity of this nucleus on the body temperature. As you can see in this movie, we use an infrared camera to measure the mouse bite temperature from above. With a very short laser pulse, we observe that a sustained increase of bite temperature. So more detailed measurement shows that activating this nucleus, neurons can increase the bite temperature, body cold temperature, even to nearly 40 degrees, and heart rate and blood pressure. On the other hand, when we inhibit this nucleus with using STGTA opsin, we found a decrease of the bite temperature, body temperature, heart rate, and blood pressure. So that seems that the activity of this preadenorphin positive VMH neurons can provide a bilateral control of this body temperature. Consistent with what we observed with cosmic signal recording, we saw a robust effect on body temperature using stimulating or inhibiting this nucleus in both sexes. One additional experiment we did is that when we based on the observation that usually stimulation of this preadenorphin neurons can induce a locomotion of the mouse. So could it be possible of the effect the body temperature was secondary to the motor function? To test this possibility, we repeat the optogenetic stimulation while using the height restraint to limit the mouse movement. Despite no observable movement, we still see a robust effect on the body temperature. There is a trend towards to smaller effect size compared to the free moving condition. That we think the motor effect partially contribute to the body temperature effect, but not a primary effect. So let me briefly summarize what we found so far. We identified the preadenorphin positive neurons in the VMH showed specific activation to the cold temperature. And the activity of this neuron control the body temperature in a homeostatic way. Since the VMH is not part of the classic thermoregulation circuits, we next ask whether the breed region could, which breed region could interact with this with it on the circuitry level. So first is we found that a local downstream target of this neuron is dorsal medial hypothalamus. We found when we adjust the placement of the fiber, we can cause a potent activation of the DMH, likely due to activation of innervating fibers in the VMH. So we tested the effect activating the VMH to DMH projection and found that activating this projection could lead to a increase of the body temperature, but to a lesser degree than the cell body activation directly. Having found that the VMH to DM projection is sufficient to promote the thermogenesis, we next ask whether this projection is necessary to mediate the effect. We implant a method with double virus injection into this hypothalamus nuclei. We use an inhibitory chemogenetic virus can inhibit the DMH, and excitatory optogenetics virus in the VMH. So using this strategy, we can suppress the DMH activity while activating the predynorphin neurons in the VMH. We found that by inhibition, the DMH can see just with the saline laser. By inhibit saline with laser means that activating the VMH, so it can cause really cell body activation, can cause a body temperature increase, but when we inhibit the DMH, can reduce the thermogenetic effect. This indicates that the projection to the DMH can be partially contribute to the effect of the VMH. We then test the effect of these cells' long-range projections. The long-range output of the predynorphin cells in the VMH are primarily enriched in the preoptic area and PEG. By activating the projection terminals in this region, we observed a potent increase in bed temperature and body temperature in both sexes. So using the retrograde AAV strategy, we label the PUA projecting and PEG projecting predynorphin positive cells separately with two different fluorescent proteins. Interesting, we found a substantial overlap in the labeling, indicating that many of these neurons projected to both regions. We further examined the inputs of the predynorphin positive neurons in the VMH using the Cre-dependent rabbit tracing. We found a wide range input of this nucleus, including many brain regions that are not considered to have function related to thermoregulation. Together, we found a predynorphin positive cells in the VMH receive both input from other brain regions and send excitatory projection to downstream regions for thermogenesis, suggesting a role of the integrate the information related to the thermoregulation. This anatomy promotes us to explore some other functions of this nucleus. One of the functions we test is related to the energy balance. We first inhibit this neuron with a chemogenetic way. We saw an increase in food intake of the mice. Then we use test the chronic effect. We inject caspase-3 and induce apoptosis. Consistent with the short-term effect, we found that after we ablate predynorphin positive neurons in the VMH, the mice gained significantly more weight than the control group. Therefore, our data indicates that these cells can be a target for the body weight regulation by modulating the energy balance. The second function we tested is related with social interaction. This was inspired by the recent publication showing that social stress could lead to a thermogenesis. We found that that's true, indeed it's true. Even a brief interaction could induce a reliable body temperature increase. You can see there's clearly a body temperature increase during the social interaction. Interestingly, we found the predynorphin positive neurons in the VMH also activated during the social interaction. Detailed analysis showed that the activation of these neurons during social interaction closely matched to the increase of the body temperature in both sexes. We then used the optogenetic inhibition of the predynorphin positive neurons in the VMH and found that social interaction induced the temperature increase was blocked. This indicates that these neurons are necessary for the thermogenesis related to the social interaction. Very curiously, we found that inhibiting this neuron leads to a reduce of the social duration interaction. This is a very interesting result, although we didn't know the exactly mechanism underlying this phenotype. In summary, our work already identified that the VMH to be a cold-sensitive region using the systematic screen by the FMI. And then we identified the pre-dynorphin positive neurons in the VMH to be a cold-sensitive neurons and demonstrate their bidirectional control of the body temperature. They are important addition to our understanding of the thermoregulation in the brain. In addition to their connection to the classic thermoregulation circuits and the control of the body temperature, we also found that they can promote the energy balance, social interaction that related to the hypothermia. Okay, in the end, I like to acknowledge the people who did the work, Chen Zhang and Ying Anxi for all the experiment, doing the experiment. I would also like to thank my collaborators, Zhifeng for his expertise in the FMI, and Xiaohong for her help with the social interaction assay. Okay, thanks. Thank you for all the listening, and I welcome any question related to this project.
Thanks so much. Awesome talk. We're open for questions. So I wonder if any of the circuits that you define now, if they eventually lead to innervation of adipose tissue to have some of their effects.
Yeah, Doctor, you mean like the predynorphin positive in VMH or not?
Maybe. I'm just wondering if there's a direct connection to adipose tissue at some point in the circuit.
We didn't do that. We injected the PRV that can be multiple ritual tracing for that. We can find the VMH got some positive neurons. But if we do the one step, like did the retrograde tracing, we only can find the RPA, some positive neurons in the brainstem, but not the VMH. So I think it not directly regulate the body temperature.
Got it. Okay. Other questions?
Joel, I will. You don't have a question? Okay. I'll quickly about the. Okay, go ahead.
So Joe, we know that during sleep, the body temperature is normally lower than wakefulness. So do you think this region also involved in physiological sleep-wake regulation?
We check the activity of this PDAM positive neuron, like predynorphin positive neurons in VMH. They are really activated during wakefulness. So that is consistent with that it can induce that increase the body temperature. But because the mice got really fragmented sleep, so we didn't see a clearly a body temperature drop during really short non-REM sleep.
And how about in the anesthesia, to induce a long-term sleep stage?
We didn't do that for the predynorphin. Did the experiment, but that is really interesting question. We also want to check whether other sleep active neurons whether can really involved in the thermoregulation as well. Because during my PhD lab, they did in the preoptic area, they found NOS1 positive neurons really regulate sleep and body temperature together.
Okay, thank you.
Evan Snyder, do you want to ask your question based on your clinical insights?
Sure. This is not. Can you hear me? Okay.
Oh yes.
This is something that you talk made me think about, and Aaron's right. This comes from the clinical perspective. Standard of care in neonatology for for babies who have sustained hypoxic ischemic brain injury is to institute 3 days of whole body hyperthermia to 33 to 34 degrees in an effort to provide neuroprotection. I was wondering whether you could speculate based on your work. I know you don't have the data, whether you could speculate whether that may actually be having some unintended damage to other circuits and things of that sort.
That's really interesting, like questions. I'm not sure whether because when we reduce the body temperature of the mice, really we cannot really keep the mice for 34, 33 degrees for 3 days. So we didn't know how to really keep the mice body temperature at that time, at that degree for a long time, and then test the functions.
Yeah, I didn't expect you to have an answer. It just provoked some concern that the standard of care that's being used to protect against brain damage may actually be having some unintended damage based on the circuits you talked about. So thank you.
Thank you.
Okay, we have a question from Jin Yun Kim. Do you want to ask it?
Do you know any?
Hi, I'm just yeah, I found very interesting. Can you explain any kind of direct projections if there is anything, because MPOA is more less sensitive typically to the temperature, ambient temperature.
Yeah,
so more of input
yeah, because we saw the projection to the we saw the VMH pre-synaptic neurons project to the preoptic area, and for the rabbit tracing, we saw also a preoptic area got some nuclear neurons project to the VMH, so it's got a reciprocal connection. So we didn't really look into how to dissect the circuit, but it's really interesting to test whether this cold sensitive neurons innervate the warm sensitive neurons in the different brain regions. Yeah, we just haven't checked.
Because that reminded some circuit from MPOA and VMH for the social mating versus the fighting behavior. So I just wondered that connections, and also whether you have seen any kind of behavior when you gave the male and female intruders for those P-norepinephrine neural activity, any correlation with their behavior for any kind of mating or fighting.
Oh, that's a really nice question. Just we didn't look, when we did the terminal activation, we didn't do any social interaction behaviors. So we don't have the answer yet. Yeah.
Thank you.
Okay, any other questions?
Hi, Joe. I have a related question about the interesting temperature going up. During social interaction. About just 1 degree, right?
Yeah, it's about 1 degree.
It seems to me interesting, kind of arousal stage. So I'm curious about first in other related aggression behavior state. Have you been able to observe any body temperature increasing moderately?
During this social interaction experiment, we used young mice, so we didn't see any mating aggression behavior in that assay. So I didn't know that, but other people did in the DMH, they found that the social stress also can increase 1 to 1.5 degree, due to the fighting or some social stress.
Yeah, that's very interesting. We should keep asking why this 1 degree body temperature increasing is so critical for behavior consequences. I have no idea.
I'm
why.
I don't have the exact mechanism, but I'm wondering whether it's because it's increased the thermogenesis for the muscle, skeletal muscle, for the next step, fighting or running something.
Okay.
Increase the
that's also explained by maybe the social behavior is one of the kind of innate behavior coding by the hardwired genetic circuits.
Yeah,
thank
maybe
you.
also like for you guys, maybe it's like the
Sure.
warm, the fuzzy feeling that it can be a social reward that promotes this behavior. Yeah.
Thank you.
Every week for NeuroZoom, I get that warm, fuzzy feeling seeing all of you. So thanks, Zhe. Awesome talk. Really a comprehensive study. That's going to be a really important paper. So congratulations. And Zilong, you're up to introduce Weiguang.
Yeah.
Okay, it's my great pleasure to introduce second speaker. The first speaker is ION faculty. The second is ION alumni. So Weiguang has got his bachelor degree from Sichuan University School of Medicine. So he was training as a physician. So after a bachelor degree, he went to ION to do his PhD with Professor Taylor Xu, where he started learning to perform the neurobiology of ion channels, particularly ASIC and sensing and ASIC channels. So after PhD training, he went to did a postdoc system investigator in School of Medicine, Shanghai Jiao Tong University, where he developed another series of interests away from his mentor, which is the we'll talk about today is the neurocircuitry underlying the memory extinction. So it's very interesting. So we're going to starting his own lab last year in the Neuro Institute of Shanghai. I hope some of you will be visiting. It's a Fudan Neuroscience Translational Institute. So also gathering a group new talents there. So today we're going to talk about the cellular synaptic mechanism underlying how we forget our fear memory forgetting. Okay, welcome, Weiguang.
Okay, thank you. Is the screen okay?
Yeah, great.
Okay. Thanks very much for the kind invitation and introduction. I'm Weiguang Li, coming from Institute for Translational Brain Research at Fudan University. It's a great honor for me to present our recent progress on fear extinction. Today's title is Cellular and Synaptic Mechanism Underlying Forgetting of Fear Extinction Memory. I received my PhD degree in neurobiology and at the Institute of Neuroscience. Then I went to Shanghai Jiao Tong University School of Medicine for postdoc training and was appointed as a dependent faculty member there in 2014. And last year, I joined Fudan University and established a laboratory entitled Memory Principles and Applications, with a long-term goal to understand the memory nature in emotion, feeling, and motor behaviors. The work reported today was done at Shanghai Jiao Tong University School of Medicine. Fear is a conserved emotion and essential for survival at the challenging environments. Excessive fear is a major cause of post-traumatic stress disorder. Fear can be learned to form a fear memory in laboratory auditory fear conditioning, in which the conditioned stimulus is associated with conditioned stimulus, such as electrical shock, represents a powerful behavior model used to study fear memory. Conditioned fear response, like freezing in rodents, can be elicited upon re-exposure to the auditory conditioned tone. Notably, fear memory can be extinguished via repetitive tongue presentation without food shock, or process termed as extinction. However, the extinction is not erasure of the original memory. The extinguished fear memory may return with the passage of time, spontaneous recovery, or following an aversive event, fear reinstatement, or in a new non-extinguished context, fear renewal. Mechanistically, fear extinction depends on plasticity of synaptic connections in the medial prefrontal cortex, basolateral amygdala, and ventral hippocampus, and other brain regions. It's well known that help plasticity constitute a fundamental cellular mechanism for memory formation and retrieval. Here, we raised our key scientific question, namely the Hab plasticity underlying fear extinction and its failure. First, how does fear extinction form its synaptic and cellular representation in the brain? Second, why is memory of fear extinction easily forgotten? Although fear extinction can be learned by repetitive training, the effects of extinction is always vulnerable. Or context-specific relapse of extinguished fear is known as fear renewal. Both appropriate context and the conditioned tone are required, but neither alone is sufficient to cause fear renewal. Typically, in a mouse with cued fear memory established in context A, repetitive presentation of the tongue leads to fear extinction in context B. However, the fear memory can be invoked when the mouse is tested with the tongue in the conditioning context, context A, or in any other context distinct from the extinction context. Due to the context dependence in fear renewal, hippocampus and related circuits like the projection from ventral hippocampus VHPC to central amygdala CA, and a feed-forward projection from VHPC to MPFC are necessary for fear renewal. While it's known that synaptic plasticity in lateral amygdala gates fear memory engram formation, it remains unknown how fear renewal happens in a memory-like manner, namely how the conditioned tone and context information work together to drive fear renewal behaviorally. Make the long story short, we identified an input associativity underlying fear renewal. The importance of input associativity in fear memory relapse was appreciated by the field and highlighted. To understand the representation of memory in single cell resolution, the memory engram cell has been proposed, defined, and identified by using diverse memory paradigm in multiple brain regions. And for a cell to be the memory engram cell, there are three criteria. First, the cells are activated by learning. Second, the cells have enduring cellular changes. And third, reactivation of these cells causes memory retrieval. Based on fear memory engram synapse, fear extinction can be understood as a reduction of fear conditioning induced enhancement of activated spines. Moreover, reconditioning restores fear extinction induced reduction of activated spines. However, the newly activated cells and connections during fear extinction, namely the engram neurons and connections for fear extinction, are largely unknown, particularly the location, distinction, directionality, and adaptation of fear extinction engrams remain to be established yet. We took advantage of the targeted recombination in active population trap system, which is developed by Professor Li Qunluo lab. Using the trap-crossed H2B flux mice, they mapped out the extinction engram neurons across the whole brain. The double transgenic mice were trained for fear conditioning and extinction, and subsequently inject IP with 4-OHT before day 4 of extinction, where when the fear response was low, resulting in the expression of H2B GFP in neurons active during extinction. Many brain regions, including MPFC, BLA, and VHPC, and several thalamic nuclei were active by retrieval of extinction memory. Reactivation of trapped cells active during extinction by memory retrieval was assessed by subsequent C-force staining, showing that the extinction engram neurons are reactivated during extinction retrieval, but suppressed by fair renew in MPFC, BLA, and VHPC. Consistently, extinction retrieval also invokes a much higher excitability and enhanced global synaptic drives to the engram neurons over the non-engram neurons. Next, we explored the function of extinction engram neurons in the regulation of memory-related behavior. We employed optogenetic inhibition to silencing the extinction engram neurons. During the extinction test, we identified the necessary roles of extinction engram neurons in MPFC, BLA, or ventral VHPC for retrieval of extinction memory. Likewise, activation of extinction engram neurons promotes the expression of extinction engram memory to counteract fair renew. In summary of the location and distinction for engram neurons, retrieval of extinction memory recruits a subset of neurons as extinction engram in the interconnected circuits composed of MPFC, BLA, and VHPC. Optogenetic inhibition or activation of extinction engram neurons suppress or promotes retrieval of extinction memory. Extinction engram neurons show much higher excitability than non-engram neurons following memory retrieval. We then evaluated the functional coupling from BLA engram to or VHPC engram to MPFC engram neurons. We found that optogenetic stimulation of BLA or VHPC extinction engram neurons at home cage increased the reactivation of engram neurons in the MPFC. On the contrary, optogenetic inhibition of BLA or VHPC engram neurons during the extinction memory retrieval reduced the reactivation of reactivation of MPFC engram neurons. With similar strategy, we also evaluate the roles of the opposite roles from MPFC to BLA or MPFC to VHPC extinction engram neurons. When the MPFC extinction engram neurons are optogenetically stimulated, reactivation of BLA or VHPC extinction engram neurons were not affected in the home cage. Optogenetic inhibition of MPFC extinction engram neurons also didn't affect the reactivation of BLA or VHPC extinction engram neurons during the extinction test. This data suggests BLA and VHPC engram neurons are functionally linked to MPFC engram neurons in a directional manner. Trans-synaptically labeling pre-synaptic neurons of MPFC engram neurons identified that MPFC engram neurons received inputs from BLA and VHPC, supporting a direct connectivity from BLA and or VHPC to MPFC engram neurons. We then manipulated the synaptic connectivity of engram neurons from BLA to MPFC to affect extinction memory retrieval. We found that silencing MPFC engram neurons led to a failure of optogenetic activation of BLA engram neurons to invoke extinction memory retrieval. Interestingly, in an opposite direction, optogenetic stimulation of MPFC engram neurons still promoted the retrieval of extinction memory despite the silencing of BLA engram neurons. With similar strategy, we also identified the directional connectivity of VHPC engram to MPFC engram. In summary of directionality between engram neurons, instead engram neurons constitute an engram construct in the term of directional engram connectivity from the BLA or VHPC to MPFC, but not that in the opposite direction for retrieval of extinction memory. Notably, extinction memory is not always retrievable, and its inhibitory impacts on file memory subside with passage of time, namely the extinguished file behavior can recover spontaneously over time. By contrast, a group of mice received additional extinction training and re-extinction the day before the memory retrieval to re-attain their fair extinguished state. Spontaneous recovery test failed to invoke the reactivation of engram neurons in MPFC, BLA, or VHPC, supporting that forgetting of extinction memory might be due to insufficient activation of the engram neurons. Consistently following the spontaneous recovery test, the extinction engram neurons showed a much lower excitability and reduced synaptic drive than the non-engram neurons. We suppose that forgetting of extinction memory might be due to insufficient activation of engram connectivity, while optogenetic stimulation of engram projection overcomes the spontaneous recovery test. We then measured the adaptive changes of BLA to MPFC and VHPC to MPFC engram connectivity in mice following spontaneous recovery or re-extinction training. In the spontaneous recovery, but not the re-extinction group, the paired pulse ratio of BLA to MPFC and VHPC to MPFC engram connectivity were increased in MPFC engram neurons compared to the non-engram neurons, suggesting that spontaneous recovery compromised the pre-synaptic release probability at these engram snaps. We also compared the AMPA to MD ratios between MPFC engram and non-engram neurons, identified the post-synaptic potentiation happened only in the engram nuance of the re-extinction group, but not the spontaneous recovery group. These results suggest spontaneous recovery may be due to suppression of engram connectivity, and that expression of extinction memory is associated with enhanced strength of engram snaps. With similar strategy, we failed to identify the synaptic adaptation of MPFC to BLA engram connectivity, consistent with the directionality of the engram snaps for retrieval of extinction memory. We then used optogenetic approach to access the extinction engram connectivity to enable extinction memory retrieval to prevent spontaneous recovery. In vivo delivery of engram specific optical long-term potentiation prevented spontaneous recovery, or behavior effects of which was equivalent to re-extinction learning. To corroborate the effects of optical RTP that enable extinction memory retrieval, we monitor the projection dependent neuronal activity of engram neurons in MPFC, the fiber photometric measurements of calcium transients to address whether engram connections of BLA to MPFC and VHPC to MPFC are potentiated in vivo after LTP induction. Indeed, as a proxy for strength of BLA to MPFC and VHPC to MPFC engram projection, calcium signals in MPFC engram neurons were enhanced after LTP induction. In summary of the main finding, we established a dynamic triple-rate construct of inter-regional engram circuits underlies forgetting of extinction memory. Let's give our answers to the scientific question raised before. Question 1, how does fear extinction form its synaptic and cellular representation in the brain? Our studies identified a new learning process involving a tripartite construct of inter-regional engram circuits. Question 2, why is the memory of fear extinction easily forgotten? Firstly, input associativity, underlying reactivation of the original fear memory engram neurons drives fear renewal. Secondly, inaccessibility of extinction memory engram neurons due to insufficient synaptic activity and analyze spontaneous recovery. Finally, thanks very much for the lab head and my PhD and postdoc advisor, Professor Tianle Xu, and the major contributors, Dr. Wang Qi, Wen Jiao, and Gu Xue, in addition to the lab members. When I worked at Shanghai Jiao Tong University, I thank my own lab members at Fudan University. I also thank our collaborators for their great contributions. Thanks for your attention. Thank you very much.
Great. Thanks, Weiguang, for a beautiful talk. Now we open for questions. Yeah, may I ask a first quick question for the, I'm curious about the role of PBT. I saw the activation in the first initial screening. So there must be play some role, right? So what's your thought about that?
Yeah, it's an interesting question. We tried to explore the role of PBT in extinction and memory. It has known. It was known that PBT has a role in temporal processing for memory before.
Is it has a direct connection with hippocampus or amygdala? I believe not, right?
Yeah, it has a directional
okay.
connection to hippocampus and amygdala. Yeah.
Okay, great. So and last question comments that you mentioned about the fear memory extinction. So how about the pleasant memory extinction? Is that will be different, right? So it had you give sugar to the mouse and had a memory extinction for a while. So it will be the similar or different memory extinction or forgetting mechanism. You get the pleasant memory versus aversive memory.
Yeah, there must have some similarity. I think the MTFC may play a major role to inhibit the previous memory, including a present memory. For forgetting, I think it's a time-dependent decay of a memory, not a new learning process. Yeah.
Great. All right. Now the questions.
May I ask a quick question?
Sure.
Okay.
Go ahead.
Hi, Professor Lee. It's a very great work. So I'm just wondering, the protocol you are using for fear extinction, are you using different tone or that is the same tone?
Same tone.
Okay. So you are using a different contact. It's just the contact your fear extinction.
Yeah,
right?
yeah. Different context. Yeah.
Okay. Yeah. Thank you.
Thank you.
Okay. Any other questions? Okay. If there are no more questions, so yeah, should we thank the Zhang and the Weiguang for great talks today?
Thank you.
Always you're there.
Thank you.
Great. Thank you. Good luck with the. All right.
Okay, thanks everyone. See everyone. Not next week. No talk next week because of
Still in 2 weeks.
Yeah, September 12th. It's 13th in China.
I'll be broadcasting from Shanghai again.
Yes.
Okay.